Sie es auch zu einem späteren Zeitpunkt anschauen können oder nochmal anschauen können, um gewisse Dinge vielleicht auch zu vertiefen. Und Sie haben auch die Möglichkeit, im Chat Fragen zu stellen, die wir dann im Anschluss an die Vorlesung, an den Vortrag beantworten werden. Heute ist es mir eine ganz besondere Freude, Ihnen Frau Professor Ulrike Protzer ankündigen zu dürfen. Sie ist ja eine der Expertinnen in Deutschland, wenn es um Viren geht. Wir haben sie schon oft sehen dürfen im Fernsehen, im Radio. Und sie hat immer brilliert durch ihre klaren und klugen und informativen Worte zur Pandemie. Und das fand ich schon sehr beeindruckend. Und wir sind sehr, sehr stolz, dass wir sie hier in unserem Kollegium bei uns haben. Ulrike Protzer ist Medizinerin. Sie hat Humanmedizin studiert, unter anderem auch in Bayern, in Erlangen, aber auch in Südafrika und hat dann ihre Ausbildung, ihre Facharztausbildung zur Inneren Medizin absolviert und dann noch eine weitere Facharztausbildung draufgesetzt mit dem Schwerpunkt Virologie, Mikrobiologie und Infektionsepidemiologie. Also eine ausgewiesene Expertin auf diesem Gebiet. Und seit 2007 ist sie hier die Direktorin des Institutes für Virologie, auch Direktorin am Helmholtz Zentrum hier in München und hat den Lehrstuhl für Virologie hier inne. Heute geht es um das Thema, wie wurde der Virus entdeckt und wie wurde die Impfung entwickelt, wie wirkt die Impfung. Und es ist genau ihr Thema, denn sie forscht nämlich darüber, wie wir Menschen auf ein Virus reagieren, also die sogenannte Virus-Wirt-Interaktion ist ihr Thema und auch, welche Abwehrmechanismen wir in Gang setzen. Und das sind natürlich Voraussetzungen dafür, um Therapien, auch Prävention im Sinne von Impfung zu entwickeln. Liebe Ulla, ich freue mich, dich jetzt ankündigen zu dürfen zu deinem Vortrag heute. Du wirst uns erzählen, wie das Virus entdeckt wurde und was es mit der Impfung auf sich hat. Wir freuen uns sehr. Du hast das Wort.
Ja, ganz herzlichen Dank, liebe Marion. Es ist mir natürlich eine große Ehre, hier heute so ein bisschen was über die Grundlagen des Virus präsentieren zu dürfen. Und während ich meinen Bildschirm teile, vielleicht nur ganz kurz, ich wurde gebeten, das Ganze auf Englisch zu halten. Deswegen werde ich jetzt also dann auch auf Englisch wechseln. Yeah, ladies and gentlemen, it's a great pleasure to present today a little bit about what we know about the SARS coronavirus, where it comes from, maybe where it goes to, and also how we can hopefully handle it. So let's start. About 1.5 years ago, I always told my students in lectures, there is a nightmare for virologists. And basically this is if a virus that is so far unknown will be there with new immunogenicity, so our immune system doesn't know it, which is highly contagious and effectively crossed the species barrier. So it can be transmitted from human to human, not only from the animal to the human, but in between the human population. And this transmission usually occurs then through droplets or aerosols, so by airborne transmission. That is what makes a virus then really efficient. And it wouldn't worry us if it wouldn't make us ill. So if there is a relatively high pathogenicity and mortality. So this was 1.5 years ago. And about 1 year ago, basically, this was there. It was the new coronavirus. And until 2019, I always recommended my students to watch a movie, which is called Contagion. And this movie really tells about a virus that is transmitted from bats to humans and causes a pandemics. And then basically now I stopped to recommend that movie, but it's still a good movie. So what are we talking about today? It's the SARS coronavirus. And this is electron microscopy pictures. Why? Because viruses are too small to be seen by a normal microscope. You need something which really has a high resolution, and that's an electron microscope. And what you see there is then, if you infect cells, then you see these little particles here, which are quite evenly formed, which look as if they would have a corona. And if you reconstruct that, then it comes out what you see here on the right side. That's the SARS coronavirus carrying these spikes on the surface, that is surface protein, which forms trimeric spikes. And that's how the virus attaches to its host cell, enters the host cell, and then replicates in the host cell. And when you try to visualize that by, again, a specific way of electron microscopy, then you get this picture here. So this is cells which have these protrusions, and out of the protrusions comes the virus. And of course, we as virologists always think that viruses are beautiful. I hope you agree when you see these pictures, but they are not only beautiful, they are also harmful. And that's what you can see when you look at the numbers. That's what we all got somehow used to in the last months, I would say. And that's how the pandemic developed. And you see clearly that we had a sharp rise in late 2020. And now numbers luckily are dropping again, at least slowly in most countries, because there were measures taken. There were lockdowns, partial or more severe, and that's how we tried to contain this virus. Maybe I can say a few words about what a virus is. A virus is not something, a virus does not live. A virus cannot replicate on its own. A virus does not have a known metabolism, and a virus cannot move by its own. I think it's very important to understand that, and that's what a virus is different from bacteria, because bacteria can grow on their own. They can even crawl. They have their own metabolism. Viruses do not have that. They are basically an extremely optimized chemical composition of proteins, sometimes lipids, that cover genetic information. And this envelope is made to basically protect the genetic information and help to transport the genetic information from cell to cell. And for a virus to replicate, it has to enter a cell. And that's very, very important to understand, because if you want to know how the virus manages to spread, as if we see here, then you have to know it's us who transport the virus. It cannot run on its own. It cannot fly on its own. It's us transporting the virus. It's us replicating the virus. And only if the virus hits the next host, it can survive. It can replicate again and try to hit the next one. That's the situation we have right now if you look at a map. And I think that's quite interesting. So the darker red the color here is, the more cases of COVID-19 we have. There's a lot of cases, as you know, in the US. There's a lot of cases in the UK. There's a lot of cases in Spain and also some other countries. But most affected countries are in the Northern Hemisphere, with some exceptions in South America, but that's probably also has political reasons. The Southern Hemisphere at the moment is much, much less affected. And there is countries which are even free of the virus. For example, New Zealand or Australia, there is just no cases coming up. And even in China, it's very, very few cases which are observed. What does that mean? The picture was different in our summertime when it was wintertime in the Southern Hemisphere. There were many more cases occurring here in the Southern Hemisphere, and very few cases, as you all remember, over summer in our hemisphere. And now in wintertime, when the days are shorter, when there is less UV light, the virus is very sensitive to UV light. You'll see why in a minute. Then we have more and more cases. So what is that virus? You already learned now what a virus is. So it's basically something dead, although we always think it's very intelligent, but that's pure evolution. But looking at what this virus is, it's a member of the family of coronaviruses. And these family of viruses has been discovered in 1931 already, with the first, at that time, animal coronavirus. And the first human coronavirus was only detected in 1966. So not that long ago, about a little more than 50 years. And right after a second one was detected. And then there was basically not a lot of new ones until 2003. And in 2003, a virus emerged which caused severe acute respiratory syndrome. And that's why it was called a SARS virus or a SARS coronavirus. That was the first one, of which we now have the second one. That's why it's called the SARS coronavirus 2. And in between those 16 years, there was another member of that family emerging. And that was the MERS coronavirus. MERS for Middle East Respiratory Syndrome coronavirus that also causes quite severe respiratory diseases, and 2 new strains of coronaviruses, which cause, like the first ones, rather mild diseases, more like, well, the normal common cold viruses. So 3 severe ones, SARS-1, MERS, and SARS-2, and then 4 ones causing more mild diseases, more common cold type diseases. And I said the very first ones were actually animal viruses, and that's where all the coronaviruses come from. They come from the animal kingdom, and from the animal kingdom, they jump the species barrier and come into humans. And if they are then within the human population, then they are spread basically this way here, by sneezing, by coughing, by producing droplets, aerosols. We'll discuss that. And that's how they spread. And this is, if you have then a severe form of the SARS coronavirus, how you can end up. And of course, nobody wants that. Now, looking into a little more detail where these coronaviruses come from, then you see most of them have something to do with bats. The NL63, one of the early ones, comes from bats directly into humans. The MERS coronavirus came from bats into camels, and from camels into humans. Then there's one which came from cows. The first SARS coronavirus also came from bats over wild cats into humans. And now the new one probably also comes from bats. And we don't know whether there is an interim host or whether they directly went into the human population. Looking at that virus in a little more detail, you see again here these corona-shaped surface of the virus, and that's where these viruses have their names from. And this virus has an unusual genome. The genome is a very long RNA, 29,600 nucleotides long. You would predict, looking at that as a molecular biologist, that must be unstable. And basically it would be if the virus would not have some repair enzymes on board, which prevent that during replication, a lot of mutations occur. And that's why the virus is relatively stable for an RNA virus. There is mutants, of course, we heard about that the last days, but for an RNA virus, that's not too many. Now, this very long RNA genome encodes for several proteins. I don't want to go in detail to, but looking at what the genome looks like and comparing that to other coronavirus genomes, you easily find the nearest neighbor. And the nearest neighbor of that coronavirus, called Wuhan, because it first was described there, is a coronavirus, again, found in bats. And it's this type of a bat, which is called a horseshoe bat, which is the host of this closest relative to the SARS-CoV-2, so probably the origin where the virus came into the human population. Now let's talk about the disease. Many people say, okay, is there anything about the SARS coronavirus, about COVID-19? Does it do anything? Yes, it does. We're used to have seasonal excess death, usually caused by influenza viruses during the flu season. The flu season is always in winter times, it usually starts somewhere in December, January, and then ends somewhere in March, early April, the latest. And looking at the last flu seasons, you see that there was an excess mortality in the 2016-17 flu season. The darker it is, that means the more excess deaths from the ones you would expect just by normal death occurring due to age. So the more excess death you have. There was a relatively heavy season in 2017-18. The 18-19 season was a little more mild. Well, maybe also because people were shocked of the 17-18 season, so there was more vaccination in 18-19. That's then also how you can keep back the virus. And there was not a lot of flu in 1920. However, right after the flu season, there was an excess mortality peak, which was very clear, and that is due to the new coronavirus, the SARS-CoV2, or the disease it causes, which is called COVID-19, so coronavirus-induced infectious disease 2019. That's where the name comes from. Now, interesting is if you look into the age distribution of the excess deaths, and it's very clear that during each influenza season, the elderly die, and also the young kids are at risk. Now here, in the last season, it's also the elderly, but not only the elderly, it's also younger ones, in which you clearly see an excess mortality, but not in kids. So kids had an excess mortality in the flu season, but not when the coronavirus comes up, which is good news. That means kids usually do not develop severe COVID-19 and do not die from the virus. But as soon as you reach the adulthood, you can have excess mortality. However, very clearly, it's very, very age dependent. And the older you are, the higher your risk is. Now, are we doing particularly good in Germany? No, we're not. That is the regional mortality throughout not all countries, but 30-something countries. And you clearly see the highest excess mortality Japan has. And the different dots are just different studies made in different regions in a country. And that's always the mean, which is shown here as a colored dot. So the highest mortality has Japan, followed by Italy, Greece, and then already Germany. Also, Germany definitely has a very good health system. Certainly, Japan has a very good health system. Still, the mortality is high in these countries. And that is because we have a relatively old population. So our average age of the population is just high. And that's true for all the European countries. And that's why we are here in the top range of the mortality due to COVID-19 countries. US basically in percentage is doing better than we do. So the percentage is, quite a bit lower than in Germany, but the population is younger. And that's the reason. And countries which do quite well is countries where you would not expect that, like Kenya, Pakistan, Algeria. That's because here, usually the population is relatively young. So they have less of the problem than we have in that respect. Now, what is the disease which the virus causes? That is, it causes very different disease symptoms and also disease manifestations. Very clearly, what it does cause is a lung disease, and that's shown here on the left. That is a normal lung where you see the alveoli and a little larger bronchioli. That's how we breathe, because there's a lot of air in. And when you now look into immunohistochemistries of an acute respiratory distress syndrome, what happens very early is that lymphocytes, immune cells, macrophages accumulate in those alveoli, and you can easily guess that it's not very easy to breathe if you have such a lot of cells in your lungs. And then if that goes on a little further, then you see that even fibers and scars are formed here in the lung, and that's also why you then may have long-term consequences. But the virus does not only hit the lung, it also has extrapulmonary manifestations, as we call it, so manifestations outside of the lung. And that can be in our brain, in our nerves, because it can infect nerves. That is why you also may lose your sense of smell and taste. It can cause thrombi, so thromboembolism, hitting either your brain via stroke, or you may have a heart attack or any other type of thrombi developing in your vessels. You've heard about that last week. And by that, it hits a lot of other organs like the kidney, the liver, the gastrointestinal tract, and even the skin. Now, how do we detect the virus? You've all heard that there is the normal thing to do is a nasopharyngeal swab, and you do a qPCR, a reverse transcription qPCR, to detect the RNA genome of the virus. And if you look at the kinetics and you look at that, how it relates to the start of symptoms, then what you observe is that you already have rising virus titers and very sharp rising titers before the onset of symptoms. And then after the onset of symptoms, your virus titers slowly but surely drop. And you first detect the virus in your nose and your throat. And afterwards, you detect it in your lung when you collect some sputum or some fluids coming from the deeper respiratory tract. And after that, then you detect the virus also in the stools. And there it can even persist for quite a while. However, this is usually dead material, so this is not infectious. What is infectious? Infectious, per definition, is if you can take the swab material and you can grow the virus in cell culture. This, of course, has to be done in a high biosafety level lab, where not only it's prevented that anything gets outside into the environment, but also the people entering this lab are protected by specific personal protection equipment. And you can grow the virus basically already before the symptoms start, and then for about a week, up to 10 days after start of symptoms. And now you may say, okay, that's the interesting period. And that's definitely true, because theoretically, only when you grow the virus, then the virus is infectious. However, in vivo, it seems even to be a little more infectious than in our cell cultures. Now, you've heard a lot about these antigen tests, which do not detect the viral genome and do not amplify, but they directly detect some of the viral proteins. And in German, they are called Antigen-Schnellteste, so fast tests. And when are these tests positive? They usually are positive when symptoms start, not before. And then they are positive for about 5 days. And then they are negative again. That is because they do not use any amplification methods. They're just less sensitive than the PCR methods or the virus cell culture method. Now you already see what could be a problem. I think the problem back here, after if you are a few days after onset of symptoms and the infectivity of the virus already wanes, that's not such a problem. But the problem is that early during infection, when the virus titer is steeply rising, you may also miss it with an antigen test. That means the antigen test has to be taken with care, and you should maintain your hygiene measures even if your antigen test is negative. PCR is more sensitive, so you also will detect those who are here in the very early phase of an infection. And why is the antigen test not positive that long? That's because your immune system develops antibodies that captures the virus, that captures the antigens, and then you cannot detect them anymore. But on the other hand, that's also good, because the antibodies obviously then are a sign of the immune response and that your body is fighting the virus. Now, what we wondered is how useful are these antigen Schnelltests? And we teamed up with our partner university hospital at LMU, and we took all samples which we could get our hands on at initial diagnosis of COVID-19 in both Munich university hospitals. In the very first sample where we detected SARS coronavirus, and which was actually the source of the diagnosis. So all 100% of those patients had confirmed COVID-19. And what came out using two different assays is that we could only detect about 60% of the samples with the antigen assay. That's the ones shown in red here. That's the positive ones. And we missed about 40%. That's the blue ones. All proven COVID-19 disease, all patients who had to be hospitalized. And that is clearly dependent with the dependent on the number of virus which is in that initial sample. That may vary because some people come earlier to the hospital, some come maybe a day later. And the higher the titer is, the more likely you are to detect the virus with a Schnelltest. But there is a significant number, which quite a decent virus titer, that's 1 million or even 10 million viruses, which you may miss. And that's of course something where you have to be careful in a high-risk setting as a hospitalist, but also when you go into an elderly care home, that you do not miss your hygiene measures, even if your antigen Schnelltest or your antigen testing is negative. That's just something to keep in mind. This is a nice thing to give some indication, but it's far from being secure that you're not infected if the test is negative. So how is the virus transmitted? We already learned a lot by, basically helping to care for the very first patients in the Basel cluster, which came up here in the Munich area. And you might remember that there was a family who was kept together in a hospital, the whole family. This was father, mother, and 3 kids. And they were all the time together. And basically the father infected himself during a business meeting by an index patient who came from China. So the index patient, after returning to China, learned that she was coronavirus positive and also developed some flu-like symptoms. And the father, only 5 days after he had the business meeting, also developed flu-like symptoms. And after him, the mother and 2 of the children developed symptoms. And that is a very nice case because here there is very sure where the infection comes from. This is the only point where the father could have been infected. And we knew that is the point where the mother got positive, and that's only 6 days. And since the mother did not have any direct contact, it must have been via the father. So that means the transmission chain was only 3 days to the father and 3 days to the mother. And then also 3 days again to the kids. And only 2 of the kids got infected. Also interesting, the youngest kid, which was still breastfed, was not infected. And the kids developed very unspecific symptoms. So they did not have flu-like symptoms like the mother had and the father had or the index patient had, but they had some vomiting, some diarrhea, some more gastrointestinal symptoms, and it was rather short symptoms which they had. So a day after they started their symptoms, I visited them and they already jumped around in their room and they were fit again. So the kids really had very few symptoms and very atypical symptoms. So now, again, the question, how is the virus transmitted? So it's transmitted via droplets. That means if you sneeze or if you cough, it is transmitted via aerosols. This is basically small droplets. And it can be transmitted by smear infection, but smear infection is not very efficient for virus transmission. Why? As I told you at the beginning, the virus does not replicate on itself. So if you, for example, sneeze and there is some virus on a surface, then the virus easily dries out and dies because it cannot replicate on its own on the surface. It's not very stable there. And if you now grab to the surface and then want to infect yourself, you basically have to take your hand and bring it into your nose or into your mouth to infect yourself. And when we wear masks, this also is prevented because there is just no way to directly touch your mouth or nose. So that's the least important way. But droplets and aerosols are important. What are droplets? What are aerosols? Droplets are larger drops at a size of more than 10 micrometer. So a millimeter is what you all know, and a micrometer is 1,000th of this. So 1% of your millimeter is a droplet, anything which is larger than that. And aerosols is basically small droplets. That's droplets where part of the liquid of the fluid is lost, is dried off, and then it's called an aerosol of a size of less than 5 micrometer. And the point is, droplets are relatively heavy, so they fall down to the bottom within 1.5 meters. That's why we say keep a distance of 1.5 meters. Aerosols don't do that. Aerosols can be in a room for quite a while, and because they are too light to fall down, they can just fly around like clouds, like waves in a room. And that's important to know. And aerosols are produced if we breathe, if we speak, and the more so if we sing. So singing is the most effective way of producing aerosols. And that's probably why choirs and also meetings in churches were a lot of sources of outbreaks, because people usually were together in closed rooms where the aerosols could really accumulate. And then by singing, they just, the persons infected in that room, they produced a lot of these aerosols, which can contain viruses on their surface. And aerosols are easily dissolved if you're outside because there's a steady airflow. But in a room, they can stand for quite a while and can also maintain their activity for quite a while. So something which is rather dangerous is an elevator, because that's a closed room. There is no airflow if you do not have the elevator door open all the time. So that's a real risk factor. I don't use any elevators anymore at the moment. I try to use the stairs. It's healthy anyway. Okay, now do those aerosols really transmit? And that was shown by an animal experiment where one infected animal was sitting in a cage at the bottom, and a second potentially recipient animal was sitting on top, and there was no direct airflow between the two cages, but there was a connection with several windings, so no droplets could go through, only aerosols got through. And all the animals where they did this experiment with, it was 5 altogether, sitting on top, were infected. And that really proved that aerosols are a source of infection. How can we prevent that? Well, basically, that's a person that is infected, producing droplets and aerosols, and that is a potential recipient. And of course, here it's very easy for the virus to be transmitted or be transported via the aerosols, via the droplets, from the donor, the infected person, to the non-infected person. Now, if the non-infected person wears a mask, that helps, definitely, if the mask is on the dense side, but it helps a lot more if the person that is infected wears a mask, because that prevents that aerosols are formed. They just aggregate to droplets, and the droplets stick to the mask. So that's very effective. And the most effective is if both persons wear masks. And which types of masks are suited here? That's also a big discussion, with all the discussion that we now have to use FFP2 masks in public rooms. Now, where would FFP2 masks are? That's the blue ones, that is procedure masks. The red dots, that's surgical masks. That's what you all know from the operation theater. So these blue ones, the more normal masks. And this is a N95 KN95 mask, which when you look at your mask, which you think are FFP2 masks, very often have a standard that's called N95. That's an American standard, not a German standard. And if the mask fulfills that standard, then very few particles can go through. If it's only an FFP2 standard, it doesn't help a lot. A lot of particles go through. Then the surgical masks are almost as good. However, looking at which size are we talking about? Droplets is more than 10 micrometer. So all the medical masks do their job. 5 micrometer aerosols, also all the medical masks do their job. Only very small particles, which usually are not there because the viruses stick to the aerosols, could be a problem in transmission with the normal medical masks. And basically, that means you do not necessarily need one of those more expensive N95 masks, but the normal medical masks also do their job. This is in contrast to community masks. So if you take a cotton community mask, that's much, much, much worse. That's the yellow curve here. So there's almost no filtration effect, or very little. There is some effect for larger particles, no question, but it's much worse than the medical masks. And if you have a gauze or a very thin one, then it's even worse. So clearly the medical masks are better than the community masks, but it's not a big difference which type of the medical masks you use. What about now stability of the virus if it's transmitted by the different ways? So if you cough, or there's aerosols, or it's on the surface. So looking at that, for both viruses, the SARS coronavirus 1 shown here in blue and the SARS coronavirus 2 shown here in red, you see that the virus titers rapidly drop on copper, on cardboard, on steel, on plastics. This is minutes here. So within an hour, there's basically no living virus on the surfaces anymore. But if you look in aerosols, and we can look a little more in detail here, then over 3 hours, the virus on aerosols only loses about 50% of its infectious titer. So very little. For 3 hours, the virus standing on aerosols maintains its infectivity. And that is why the aerosols you cannot see are so dangerous. And this is why we always say, open the window, have airflow, have fresh air going in, and obviously also wear your masks. Now coming to the last point of my lecture today, the vaccine. Let's again have a short look at the SARS coronavirus. We already said it's this large RNA genome, which is protected by a capsid that's directly attached to the genome, and then by an envelope, that's a lipid envelope. And in that lipid envelope, you find the surface proteins, in that case, the spike proteins here, which is the most important ones, and also the envelope proteins down here. But the important ones, which also make the contact to the receptor, are the spike proteins. And looking in detail at the spike protein, it consists out of 2 types of proteins, the S1 protein shown in orange and the S2 protein shown in brown here. And within the S1 protein, there's a domain which is called the receptor binding domain, or RBD. This is where the virus directly attaches to its receptor. That's how it infects. Now, if you want to generate a vaccine, you obviously want to prevent infection. So what you want to do, you want to prevent that this spike protein here can make contact to the receptor. So what you need is antibodies, which are formed by your immune system, and that would bind to this surface part of the virus and would cover it. And then the virus cannot attach to the receptor and cannot infect anymore. And these types of antibodies we call neutralizing antibodies. Now the question is, how can we induce such neutralizing antibodies? And basically, there is quite a few ways how you can design a vaccine. So one would be that you grow the virus in cell culture, then inactivate the virus and use the dead particles. So the virus, which is it's dead, but the non-infectious virus, which you inactivated chemically as a vaccine. That is done. And that's the Chinese type of vaccine, which we know from Sinovac, which is already approved and used in vaccination. You could also have a live attenuated vaccine. That's for measles or rubella. We have that, but we don't have that for a SARS coronavirus. It would probably also be too dangerous. But what you could do, you could simply try to recombinantly express the surface proteins of the virus, either the complete spike, so the S1 and S2 proteins, so that they can assemble and form exactly the same structure and induce immune responses, or you use only the RBD, so only the region where the virus attaches to the receptor, and express that as a recombinant protein. Both of those approaches are done. Then if you have a protein, it's never as effective on its own. It's mostly very effective, and if it forms sort of a nanoparticle or a virus-like particle, and then it's on the surface of this virus-like particle, but there is no virus gene or anything. So that's a recombinant particle. That's also a very attractive approach that's also followed, but it just took in development a little longer than what we have at the moment, which is either an adenovirus vector vaccine, that's shown here. That means an adenovirus, or out of an adenovirus, people took out part of its genome and inserted instead the spike gene of the SARS coronavirus. So that adenovirus vector from the outside still looks like an adenovirus, so still infects cells, but in the cell cannot replicate anymore, but is genetically engineered to only be able to express that spike protein as a protein. And you can even make it simpler and just use a messenger RNA. The messenger RNA is the molecule, which is the messenger molecule in a cell, and the cytoplasm in a cell, telling the cell to produce a protein. So it's a natural molecule made in thousands of copies by each cell every day, and is the messenger which tells the cell which proteins to produce. It's not a genetic information as we encode on our DNA, but it's really a messenger molecule. And people took that exact type of a molecule, encapsulated that in lipid particles to be able to use it as a vaccine. And that's the vaccine we have at the moment. Now, if we have a more closer look here, we have the mRNA vaccines you all have heard about in the meantime. So a messenger RNA stabilized version engulfed into a lipid particle. And there's basically different companies who follow that. That's Moderna, that has been approved second in Germany. Then there's the BioNTech one, the German origin vaccine developed together with Pfizer, an American pharmaceutical company. That was the first one approved. And then CureVac, that's also a German company located in Tübingen. And they're also developing an mRNA-based vaccine that will also is in phase 3 trials, but we don't know results yet. But those two have very, very promising results. That's why they were approved. The next one, the next series of vaccines is these vector-based vaccines. Usually that's adenoviral vectors because they're easiest to handle. And AstraZeneca, together with the University of Oxford, developed such an adenovirus vector based on a chimp adenovirus. Why a chimp adenovirus? Because they wanted to prevent that we have pre-existing immunity against the vector, because then the vaccine works better. And that's also approved. You've heard about that. That's widely used in the UK. Then there is one also in phase 3 trials, also going here in the University Hospital. That's an adeno-26 type of vaccine, a very well-established platform developed by Johnson & Johnson. And then there was the Russian vaccine, Sputnik 5, which was the first one which Putin gave to his daughter without any clinical trials, but they just used it. So there is an emergency use allowance, but not in Germany, Europe, or the US, only in Russia and some other more communistic countries. And then, of course, people are also developing more classical vaccine types. So the inactivated vaccines, where you grow the virus and inactivate it and use it as a vaccine. And one of that is the Sinovac vaccine. They all come from China. The Sinovac one is also approved and is used in vaccination. And then protein-based vaccines, there is no one approved so far, but that would also be a very established way of vaccines. And there was a big hope that the vaccine from Sanofi and GSK would make it, but they somehow had problems with their vaccine design. The adjuvant which they used did not prove efficacious, so they had to go all the way back, and they're still back in phase 1-2 trials now. So they're really lacking behind. One which is a little further is that from the company Novavax, that's already in phase 3 trials, so very late clinical trials, and will probably be available quite soon as well. Now let's have a little closer look into the BioNTech vaccine. So you all heard that there was an emergency use granted by first Great Britain in December 2nd, and then by the US FDA on December 10. And in Europe, it also approved now, but not for emergency use, but for regular use, because in Europe, we don't do that. We always do full approval and do not give these emergency use authorizations like the US or Britain does. That's just a different way of regulation. We are a little more on the conservative side here. Now, what does that vaccine do? It's basically 30 microgram of an mRNA, so this messenger type of RNA that is modified to be a little more stable and less immunogenic. And that encodes the full spike protein with, in that case, in a membrane-bound form and a pre-fusion conformation. So that would exactly induce the antibodies, and the antibodies shown here in violet, which we want to have, the ones which block this protein and prevent the virus from infection. Now, this mRNA is encapsulated into a lipid nanoparticles made out of a lot of different lipids. I'll show you in a minute what it is. And these lipids fuse with the cell membrane and deliver the mRNA into the cell. There it goes to the ribosome and is translated into the protein, which then induces immunity. Now, what is in that vaccine in addition to the mRNA? So like if you would look into a cornflakes package here, looking at the active ingredients, that's the mRNA, which I told you about. And then there is the lipids, which encapsulate it, and that's 4 types of lipids, 2 modified lipids and 2 natural lipids. And there's also salts in there, and also some sucrose, so some sugar to stabilize the whole thing. That's all. There's no adjuvant or nothing else in the vaccine. Now, the vaccine worked very well in clinical trials that went through the press already. There was a trial which is still ongoing, so there's still higher numbers of persons included in that vaccine, but it had reached its clinical significance, and that's why it was published and then used as a basis to approve the vaccine. And there were 21,669 vaccinees and 21,686 persons who received placebo. And in that group, in the placebo group, 162 persons developed symptomatic COVID-19 vaccine, COVID-19 disease, and were infected by the virus, obviously, and then developed the disease, and only 8 in the vaccination group. And looking at severe disease, so that patients who had to go to intensity care ward, there were 9 in the placebo group and only 1 in the vaccinee group. That means if you take this 162 out of 170 altogether who were infected, you have a vaccine efficacy of 95%. And looking on a time scale, that is the first dose here given on day 0, and a second dose then giving between day 21 and day 42. And already after the first dose, you can see that vaccine efficacy starts because there is less infection in the vaccination groups as there is in the placebo group. And while in the placebo group, the infections continue, in the vaccinee group, basically after the second vaccination, there was almost no infection anymore. So extremely few infections overall, showing the high efficacy of that vaccine. But nothing comes without a price. There's, of course, also some side effects, as almost every vaccine has, but this is no severe side effects, so nothing which lasts longer. It's all what we say a reactogenicity, so short-term effects, and mainly effects which do not only occur in the vaccinee group, but also in the placebo group. That's also something to know. It's quite interesting. It's always the vaccinee group and the placebo group shown next to each other, except local events. So some aching or a swelling of the arm, that's definitely more frequent in the vaccine group. There's also a little more side effects like headache or fatigue or elevated temperatures, but these are mild. So that's up to 38.4 degree or up to 38.9 degree in the blue columns in the vaccination group. But there's also fatigue, headache, and chills, of course, as you usually have in the placebo groups. Also, muscle pain is something which is observed. So you feel a little bit like if you would acquire a cold. That's the typical symptoms if your body reacts to something, if your immune system reacts, and that's the reactogenicity, which you may feel. And that's, well, quite a few of the participants in the trial felt those rather mild, but still side effects. The good news is there were no severe side effects in none of the trials, not in the BioNTech trial and also not in the Moderna trial, which I don't show here because the results are almost identical. Now, very last thought, what can we expect from such a vaccine? Can we reach herd immunity and stop the whole pandemics? And basically, you would have to know how much of immunity you need to reach herd immunity. And that would be for the virus we have at the moment, about 65% of immunity. Now we have a vaccine which has 95% efficacy. Then you would have to know how much of immunity from previous infections do you already have? So how many natural immunity? And that would be something between 0 and 25% over the world. In Germany, we are rather between 0 and 5. And the rest has to be done by the vaccine. So that means looking at that with a 95% efficient vaccine, you have to reach between 50 and 65% or 45 and 65% of patients, depending on how much pre-existing immunity you have. And if we say, okay, 63% and we have 2 to 3% of pre-existing immunity, we would have to reach a vaccination coverage of about 60%. Well, that could be doable, but one point is a little more problematic. That's that we do not vaccinate children. So these 60% have to be reached all in adults. And that means it's depending on the age distribution of a population between 58 and 85%. So in Germany, between 65 and 85% of the population, we will have to vaccinate before we really reach herd immunity and the virus cannot spread anymore. That will be hard to reach looking at the numbers that have been already vaccinated, and that's the numbers of today. So most vaccinations have been done in Israel. That's already 33% of the population which has been vaccinated. The United Arab Emirates also have vaccinated more than 20%. They use the Chinese vaccine. The next one is Bahrain, then the United Kingdom, then the United States, and then Germany. So we're not that bad in an international comparison with having almost 2% of our population already vaccinated. But what is maybe a little worrying, if you look at the world map again, then you see it's basically the rich countries which have started vaccination. In the US, in Europe, Russia started, and China. and the less developed countries, they did not even think about starting the vaccine so far. So that's also a point we have to solve at the end to stop the pandemics probably. Now that's brings me to the end. And basically, I told you a lot about the different layers of protection we try to establish, and somebody picked it that as a Swiss cheese respiratory virus pandemic defense. And the last layer now we have is the vaccine. And with that, I'm at the end, and thank you very much for your attention.
Ja, liebe Ulrike, vielen Dank für deinen tollen und klaren Vortrag und auch für die vielen praktischen Tipps. Du hast, finde ich, sehr, sehr gut erklärt, was die Masken können und wie sich das Virus durch die Aerosole verbreitet. Deswegen ist es ja auch gut, immer wieder mal zu lüften, dass die Aerosole nach draußen können. Und du hast uns auch deinen privaten Tipp ganz verraten, dass du lieber die Treppe nimmst. Das kann ich nur unterschreiben, ist so ohnehin gesünder als in so einem geschlossenen Raum, der ja kaum einen Luftzug erfährt, wie der Aufzug rauf und runter zu fahren, wo sich das Virus im Aerosol lange Zeit hält. Vielleicht auch noch der andere Tipp, der mir gut gefallen hat, war der Film. Der ist wirklich sehr, sehr zu empfehlen. Also wer ihn nicht gesehen hat, der sollte sich ihn anschauen. Jude Law spielt auch mit, also wirklich klasse. Ja, es sind ganz, ganz viele Fragen gekommen. Ich hoffe, du hast noch ein bisschen Zeit für uns. Wirklich ganz tolle Fragen. Ach Gott, jetzt kamen noch mehr. Gott, um Gottes Willen. Ja, ich glaube, hier ist eine ganz kluge Frage, die würde ich gerne zuerst mal vorlesen. Wir haben ja eine Impfstoffknappheit nach wie vor. Wir haben nicht genügend Impfstoff in Deutschland. Deswegen hier ist eine kluge Frage. Ein Zuschauer schreibt, dann ist nach durchgemachter Infektion nicht von einer ähnlichen Immunität auszugehen wie nach einer Impfung. Wenn es so wenig Impfstoff gibt, warum bestimmt man nicht vor Impfung die Antikörper im Blut, um Personen zum Beispiel mit asymptomatisch durchgemachter Infektion zu identifizieren und so Impfstoff zu sparen? Was sagst du dazu? Ist das eine kluge Strategie?
Also das ist sicherlich so, dass jemand, der weiß, dass er eine Infektion durchgemacht hat, momentan in der Situation noch keine klare Impfempfehlung hat. Aber wir haben eine große Studie auch selber hier gemacht, wo wir alle Mitarbeiter des Klinikums eingeladen haben, teilzunehmen. Und erstens sind es natürlich nicht viele, die eine Infektion bisher durchgemacht haben. Also selbst hier in München, wo wir eine heftige erste Welle hatten, sind es nur 2% gewesen vor Beginn des Winters. In anderen Regionen Deutschlands viel weniger. Und zweitens verliert man auch nach der Infektion seine Antikörper wieder. Also nach 8 Monaten, wo wir jetzt Beobachtungszeit haben, haben 25% ihrer Antikörper schon wieder verloren. Und deswegen sagt man eigentlich im Moment, diejenigen, die es wissen, die sollten es nicht machen. Aber grundsätzlich bei jedem Antikörper zu bestimmen, wäre einfach auch logistisch zu aufwendig und würde dann noch länger dauern, bis wir die Vakzine wirklich zu den Menschen bringen. Deswegen hat man sich da dagegen entschieden.
Mhm. Eine weitere Frage zur Impfung. Wenn jemand geimpft wurde, kann er dann das Virus noch an andere weitergeben oder nicht? Oder wissen wir das noch nicht? Oder ist es zu vermuten, dass es wie bei der Influenza ist? Oder was würdest du als Expertin dazu sagen?
Ja, also wir wissen es noch nicht. In einer der beiden großen Studien ist das tatsächlich ein zweiter Studienendpunkt. Der ist aber noch nicht erreicht, weil das natürlich viel schwieriger und viel, viel länger dauert, bis man weiß, ob noch weitere Übertragung stattfindet. Aber ich bin mir nicht sicher, ob man das wirklich hinkriegen kann, dass man so komplett steril ist, also ganz 100% sicher kein Virus mehr hat. Denn wenn man sich die präklinischen Makakenstudien anschaut, dann ist es sowohl nach einer natürlichen Infektion als auch nach einer Impfung so, wenn man die dann rechallenged, das heißt wieder dem Virus aussetzt, dann haben die schon noch ein bisschen Virusvermehrung im Nasenrachenraum. Das geht nicht mehr in die Lunge und die werden auch nicht krank. Also davor ist man gut geschützt, das sieht man ja auch. Aber so ein ganz klein bisschen Virus haben die trotzdem noch im Rachen. Das ist aber auch nur eine ganz kurze Zeit. Und deswegen, selbst beim Menschen, wenn das jetzt ein kurzer Zeitraum, vielleicht so 24 Stunden ist, dann bin ich jetzt nicht 100% sicher, dass ich es nicht mehr übertrage. Aber die Übertragung ist sicherlich trotzdem deutlich.
Sicherlich deutlich geringer zu erwarten. Mhm. Gut. Dann eine Frage zur FFP2-Maske. Warum ist es jetzt sinnvoll, diese zu tragen?
Da müssen Sie Herrn Söder fragen.
Okay, gut, alles klar.
Ich persönlich glaube, dass es ein medizinischer Mund-Nasenschutz ganz genauso tut.
Genau, also du hast
alle,
es sehr schön gezeigt.
Alle klinischen Daten sagen das auch. Und
naja,
ich glaube, da muss jeder, wenn sich jemand sicherer fühlt, wenn er eine FFP2-Maske anhat, dann ist es gut. Man sollte im Idealfall dann darauf achten, dass sie wirklich auch diesen N95-Standard hat, weil dann ist sie tatsächlich zumindest besser als die normalen FFP2-Masken. Aber eigentlich sind wirklich diese medizinischen Mund-Nasenschützer schon ein echtes High-Quality-Produkt und die unterfallen halt auch unter das Medizinproduktegesetz. Das heißt, die sind auch kontrolliert von guter Qualität, was FFP2 halt leider nicht hält.
Okay.
Es gibt auch sehr unterschiedliche Qualitäten, da kann man für viel Geld ziemlichen Schrott kaufen.
Also vielen Dank für deine klaren Worte. Ich glaube, das war eine dringende Frage, die viele Zuhörer und Zuschauer interessiert hat. Ja, jetzt noch eine ganz andere Frage, die auch, glaube ich, ganz wichtig ist und viele interessiert. Du hast ja gezeigt, dass es mehrere Coronaviren gibt, auch schon mehrere Corona-Arten, Viren auf den Menschen übertragen wird. Hier kommt jetzt die Frage, kann der SARS-CoV-2-Test auch positiv auf andere Coronaviren reagieren, sodass letztlich in Bezug auf das SARS-CoV-2 ein falsch positives Ergebnis resultiert, oder ist der spezifisch?
Die PCR ist hochspezifisch. Die kann nicht auf andere Coronaviren reagieren. So ein Antigen-Schnelltest, das ist wieder ein bisschen von der Qualität abhängig, kann das schon theoretisch mal tun. Also da kann man auch mal einen falsch positiven dabei haben.
So, hier kommt jetzt eine Frage für die Infektionsepidemiologin. Können Sie bitte den Zusammenhang erklären zwischen der 7-Tage-Inzidenz und der Reproduktionszahl? Wenn die Reproduktionszahl unter 1 liegt, wie können dann so hohe 7-Tage-Inzidenzwerte entstehen?
Also die Reproduktionszahl sagt ja schlicht und einfach, wie viele Menschen steckt ein Infizierter im Durchschnitt an. Die Basisreproduktionszahl dieses Virus liegt irgendwo um die 3 herum. Also wenn ich nichts mache, dann steckt 1 oder 3 neue an. Das heißt, dann gibt es sehr schnell ein exponentielles Wachstum. Wenn ich aber natürlich was mache, also Abstand halte, Maske tragen, Kontakte einschränke, dann sinkt diese Reproduktionszahl. Und wenn diese Reproduktionszahl unter 1 ist, dann sinkt auch die Zahl der Infizierten. Und zwar relativ schnell. Also wenn ich konstant eine Reproduktionszahl unter 0 habe, dann geht auch diese 7-Tage-Inzidenz runter.
Gut. Danke. Dann noch eine Frage zu Neuseeland. Die haben es ja geschafft, Corona-frei zu sein oder Covid-19-frei zu sein. Wie haben die das geschafft, Ulrike? Was meinst du?
Kontaktbeschränkung und Lockdowns. Also ich habe gerade jemanden angeworben aus Melbourne, aus Australien. Die sind jetzt auch im Moment Corona-frei. Die haben das ganz genauso gemacht. Die sind durch 2 heftige Lockdowns gegangen, wo sie wirklich nicht aus dem Haus durften, nur eine SMS schreiben für 6 oder 8 Wochen, einmal am Tag für eine halbe Stunde rausgehen. Ansonsten mussten sie zu Hause bleiben und durften höchstens in begründeten Fällen eben zur Arbeit gehen. Jeder, der nach Australien einreist im Moment, muss 14 Tage in Quarantäne in ein Hotel. Das wird vorbestimmt, allein in einem Zimmer. Da kriegt er Frühstück gereicht und aus diesem Zimmer geht er 14 Tage nicht raus. Das zahlt er selber. Der Spaß kostet etwa $4.000. Das führt dazu, dass im Moment 240.000 Australier, die eigentlich ganz gerne nach Hause wollen würden, nicht können, weil nämlich diese Hotelkapazitäten auch limitiert sind. Also das hat schon relativ drastische Maßnahmen, die gemacht haben, aber das nützt.
Ja, also das wäre vielleicht auch noch etwas, wo Deutschland nachbessern könnte. Ich habe gerade eine Mitarbeiterin von mir, die hat Familie in Moskau und die kam aus Moskau zurück. Sie selbst hat Antikörper, sie hatte die Corona-Infektion schon hinter sich, aber sie sagte, die ganzen Mitreisenden wurden nicht überprüft, keiner wurde getestet, keiner wurde nachverfolgt. Und also da glaube ich, könnte Deutschland vielleicht noch besser werden. Was meinst du?
Ja, wir sind natürlich in einer anderen Situation. Wir sind mitten in Europa. Wir sind nicht Australien und auf einer Insel oder Neuseeland, weit weg von allem anderen. Und ich weiß auch nicht, ob wir uns politisch zu solchen Maßnahmen durchsetzen würden. Wir haben ja doch ein anderes Selbstverständnis von Reisen, von freien, von offenen Grenzen. Uns ist natürlich Dimensionen schwieriger, als es in Australien oder so ist.
Aber das erklärt natürlich dann auch, warum es diesen Ländern gelungen ist. Ich glaube, das interessiert schon viele. Weißt du zufällig, wie lange der Lockdown jeweils war in Neuseeland und in Australien, dieser strenge? Weil wir haben ja jetzt gehen jetzt auch nochmal in die Verlängerung, was, glaube ich, auch notwendig ist. Und viele sind ja da schon ein bisschen mürrisch und verlieren so ein bisschen den Atem.
Also der ganz strenge Lockdown mit wirklich gar keinen, also kompletter Ausgangsbeschränkung, wo ich niemanden mehr keine anderen Familien treffen durfte und eben nur die halbe Stunde am Tag raus durfte, war 6 Wochen. Schon relativ lang.
Mhm. Gut, dann eine Frage zu den Mutationen der Viren. Was hat es mit denen auf sich? Wirken da, wenn man geimpft ist, ist man dann auch vor diesen Mutationen geschützt? Das ist hier eine Frage, die ein Zuhörer uns stellt.
Ziemlich sicher ja, weil diese Mutationen natürlich einen ganz kleinen Teil dieses Spike-Proteins beeinflusst. Und für die britische Variante gibt es auch schon Daten. Ich war letzte Woche in einem Keystone-Meeting, da war auch die BioNTech-Leute da. Die haben das schon getestet mit Serien von Geimpften, die gibt es ja, und die neutralisieren diese neue Variante sehr gut. Und davon würde ich auch eigentlich bei allen Varianten ausgehen.
Dann eine weitere Frage zur Impfung. Es ist ja so, wenn man die erste Impfung bekommen hat, sollte die zweite Impfung, der Booster, nach 3 Wochen gegeben werden. Besteht da ein Risiko für eine Person, wenn sie später geimpft wird? Es wurde ja hier auch schon diskutiert, ob man durch die Impfstoffknappheit vielleicht besser die zweite Dosis später geben sollte oder kann. Was sagst du dazu?
Also es gibt diesen Spielraum zwischen 3 und 6 Wochen für den BioNTech-Impfstoff. So ist es auch in den Studien getestet worden, so ist auch die Zulassung. Aber mehr Spielraum gibt es nicht. Wenn man das jetzt machen würde, dann wäre man außerhalb der klinischen Studienlage und außerhalb der Zulassung. Und ich würde das auch nicht machen, denn diese Zeiträume wurden einfach schon in den präklinischen Experimenten gezeigt, dass die notwendig und sinnvoll sind. Es gibt aber eine Ausnahme, das ist dieser AstraZeneca-Impfstoff. Da ist es tatsächlich in einer Substudie der Universität von Oxford so, dass es sogar besser ist, wenn man länger wartet, also 12 Wochen wartet. Das betrifft aber nicht die mRNA-Impfstoffe, sondern eben diesen adenovektorbasierten AstraZeneca-Impfstoff.
Dann noch eine Frage, wenn der Virus an der Oberfläche ist irgendwo, wie lange lebt der? Oder halt, wie lange ist er infektiös, sagen wir es mal so?
Ich hatte ja da Daten gezeigt, also weniger als eine Stunde. Es geht ziemlich schnell, auf manchen Oberflächen nur ein paar Minuten.
Und warum ist das Virus so UV-Strahlen empfindlich?
Naja, das ist ein ganz langer RNA-Strang von ja 30.000 Basen. Und UV-Licht, wissen wir, macht in den Basen Dimere und es muss nur eine Stelle treffen und dann ist das Virus schon tot. Deswegen, weil es halt dieses lange Genom hat, ist es einfach so empfindlich.
Dann noch eine ganz wichtige Frage. Wie lange hält denn die Immunität aus der Impfung an?
Das weiß man natürlich noch nicht, weil so lange gibt es ja noch keine Geimpften. Aus den frühen Phase 1-2-Studien weiß man inzwischen, dass sie mindestens 6 Monate anhält. Bei einigen gibt es schon 8 Monate Daten. Das weiß man, aber länger weiß man halt nicht, weil es länger noch keine Geimpften gibt. Aber für die Zeit haben die Antikörpertiter ziemlich konstant gehalten. Besser als bei den natürlichen Infektionen.
Wird es dann aus deiner Sicht wahrscheinlich sein, dass man sich jedes Jahr gegen Corona impfen lassen sollte oder muss, wie bei der Influenza? Oder was ist da deine Einschätzung?
Also das wäre sozusagen das Worst-Case-Szenario, dass es nach 12 Monaten weg wäre, die Immunantwort. Wenn sich diese ersten Daten bestätigen, dann sieht es aber schon so aus, als ob es eher mindestens 2 Jahre hält. Aber das ist jetzt eine Extrapolation, die kann man dann tatsächlich auch erst machen, wenn man wirklich den Zeitraum und auch genügend Menschen im Durchschnitt beobachtet hat. Aber im schlimmsten Fall müsste man es halt zu der Influenza-Vakzine dazu tun.
So, und jetzt für dich die letzte Frage. Ich glaube, wir haben dann lange genug diskutiert, aber eine sehr wichtige Frage natürlich. Was glaubst du, deiner Einschätzung nach, wann werden wir wieder ein normales Leben führen können in Deutschland?
Tja, da können wir jetzt Wetten abschließen. Wir haben gerade heute heftig diskutiert mit anderen Virologen. Ich habe das immer gesagt, ich glaube das immer noch. Das einsetzende Frühjahr wird uns letztendlich dann helfen. Also Mitte, Ende März wird die Situation deutlich besser. Und bis dahin haben wir auch die ganzen Älteren geschützt, wenn wir so weiter impfen. Also ich glaube, dann gehen wir wieder in einen entspannten Sommer. Dann dürfen wir das nur den Sommer nicht verschlafen und sagen, okay, es ist ja alles gut und warten, bis der nächste Herbst kommt, sondern dann sollten wir schon konsequent weiter impfen und sodass wir dann gut gerüstet in den nächsten Herbst gehen können.
Super. Das ist doch wirklich ein guter Ausblick. Und dann freuen wir uns auf die Sonne und auf die UV-Strahlen, weil wir ja gelernt haben, dass sie uns dabei helfen. Gut, nochmal ganz, ganz herzlichen Dank, liebe Ulla, für deinen tollen Vortrag und für die klaren Antworten, wie wir es von dir kennen. Dafür ganz herzlichen Dank. Ich hoffe, wir sehen uns alle wieder nächsten Mittwoch, gleiche Zeit. Nächsten Mittwoch ganz interessantes Thema mit der Vorsitzenden unseres Nationalen Ethikrats und unserer Kollegin Frau Professor Alina Büchs. Da geht es auch nochmal um ein ganz, ganz aktuelles und interessantes Thema, nämlich der Verteilungsgerechtigkeit. Und da sind wir ja mittendrin in der Impfkampagne mit diesem Thema. Ich freue mich auf Sie und ich hoffe, Sie sind nächsten Mittwochabend wieder dabei. Tschüss.